peptide generation. So
Thank you.
please.
Good morning. I'd like to thank the organizers for this invitation. We're going to have a slight change of gears. What I'm going to try to talk to you today, at least the biological phenomenon is described here. You have an immune cell recognizing a cancer or infected cell and killing it. One of the main things that we're interested in our lab is to understand how can the immune cells recognize aberrant or infected cells. And this is done very specifically, and it's very fortunate for us that it is done. This phenomenon shown here is extremely common in our body. It happens every second of every day, and it's very important for keeping us healthy. So let's look with a little bit more detail on the molecular interactions that make this recognition possible. On the left, you have a T cell recognizing a cancer cell, and you can see that it forms a large interface called the immunological synapse. And in that interface, on the top right, you can see a series of molecular interactions between the two cells that drive specificity of recognition. Probably the most important of those interactions is shown on the bottom right, which is the interaction between the T cell receptor, shown in red, and the major histocompatibility molecule, shown in green. In the middle of that interaction is a small peptide, shown in yellow here, which is called the antigenic peptide. One of the things that I'm very interested research-wise is to understand how these antigenic peptides are produced inside the cell. One of the reasons this interaction has become very popular during the last few years is its role in cancer immunotherapy. I'm not going to spend much time on that, just to point it out. Tumor cells are essentially afraid of this interaction because it's one of the major ways that the immune system can recognize them and kill them. So what they do is that they overexpress other ligands on their surface, like the immune checkpoint ligands on here, that essentially nullify the antigenic interaction that I just showed you and instruct the immune system to ignore them. This discovery resulted to the Nobel Prize in Physiology and Medicine in 2018 to Drs. Allison and Honjo. So how are these antigenic peptides generated? So this is sort of an overview of the pathway. It's a complicated pathway with many nodes. I will try to simplify it for today, which is my favorite way of viewing these pathways, with a cartoon. Here you have a cell. The cells have a lot of proteins, and these proteins are sampled and degraded by the proteasome to small peptides, and a few of them are transported inside the ER by a specialized transporter called TAP. There, they're further processed by aminopeptidases shown here as a green piranha, the IRAP, and they're loaded into freshly folded, nascent MHC class I molecules. These are moved to the surface and interrogated by T cells, in particular CD8 positive T cells shown here. So you have a cell that is either infected by, let's say, a virus or transformed to be cancerous. What it does have is has new proteins. These can either be mutated version of normal proteins or completely new viral proteins or oncogenes, for example. These are also sampled by the proteasome. These give rise to different peptides, which can be antigenic peptides. These are processed in the ER and then moved to the cell surface, and now the T cells can see new, novel, unknown peptides, which signify that there's something wrong with the cell. And this starts a cascade of recognition that usually leads to the lysis of the cells through apoptosis. So for today, I'm going to focus on this enzyme here that I'm showing on the left, and its role and particular molecular mechanism that it uses to generate antigenic peptides. So there's actually 3 aminopeptidases that play roles in this particular pathway of antigen presentation. I list them here. They're called IRAP1, from ER aminopeptidase 1, IRAP2, and IRAP from insulin-regulated aminopeptidase. They were all discovered to play important roles in the immune response in the decade between 2000 and 2010. So I'm going to focus on the first one, IRAP1. And I'm just going to show you just a couple of slides with biological background to try to persuade you why it's important immunologically. This is one of the earliest experiments in the field by the lab of Neil Absastry in California, who showed that when he transferred cells from mice that were knockout for this enzyme to regular mice, these mice elicited very strong immunological responses against those cells, which indicated that this enzyme could regulate how the immunogenicity of cells. Now we understand this a little bit better, and we know that this enzyme affects the so-called immunopeptidome. The immunopeptidome is essentially the sum of peptides that are presented through this pathway on the surface of cells. And this is a study from our lab a couple of years ago, where we used an inhibitor we developed for one of those enzymes to show that when cells, these are cancer cells, when they're treated with this inhibitor, suddenly they present on the surface a lot of new and previously unpresented peptides, which have the potential to be immunogenic. One of the reasons we do that is because we feel that inhibiting those enzymes can enhance the immunogenicity of cancer, and thus they can inhibitors of those enzymes can have applications in cancer immunotherapy. And these are some unpublished data from our group. This is a syngeneic tumor model where we saw that by injecting an inhibitor for one of those enzymes can retard tumor growth, which, however, can restart after the inhibitor is cleared from the system, probably because of a reset of the immunopeptidome. What I want to focus today, though, is the mechanism and the function of this enzyme. So what this enzyme does. So this is an aminopeptidase, which means that it takes peptides and trims them from their N-terminus and makes them shorter. This is important because MHC molecules require a certain length of peptides. And actually this enzyme somehow seems to know what is the appropriate length, and will trim peptide with preference for length. It will actually prefer to trim peptides that are 9 amino acids or longer, down to about 9 amino acids. Also, and this at least initially was quite unusual for an aminopeptidase, although it has specificity for the amino terminus, it seems that it has very strong specificity for other positions in the peptide sequence. The figure on the right shows kind of an extreme example. This is an artificially designed peptide to illustrate this effect. Both peptides have the same N-terminus, but their trimming rate is tens of thousands fold different. So we now know the structure of this enzyme, and it has been crystallized several times now in at least 2 conformations. One, which is shown on the left, is a more open conformation, has a large cavity in the middle that leads to the active site, and the one on the right is a closed conformation, which turns out is actually the more active one. In the middle is a morph showing how we think these 2 conformations interact with each other. This internal cavity is quite unusual and quite large, and when we first saw it, we hypothesized that the reason it's so large is because it allows the enzyme to accommodate large peptides. It also has a very negative electrostatic potential, which fits with the specificity of this enzyme for at least some of its substrates to have some positive charges. So the main questions we wanted to address with this work is, how does this enzyme, ERAP1, recognize its substrate? How can it differentiate between different peptides that have different lengths? And what is the basis for its broad sequence preferences? To try to address this, we wanted to solve crystal structures of the enzyme with peptides bound in its cavity. Of course, because we want to use the active enzyme, we couldn't use regular peptides because they will be digested. So we use these technology that these are transition state analogs. They're phosphinic peptides. They have very high affinity for the active site, being transition state analogs, and they allow us to use them as tools to investigate peptide-protein interactions. So these are shown here, two of them, a 15-mer and a 10-mer. They have a higher affinity than the warhead, the phosphinic warhead used to target the active site, which indicates that the whole peptide can have additional interactions with the enzyme. So a couple of years ago, we solved two crystal structures of ERAP1 in complex with these two peptide analogs that are shown here. In both cases, the peptide was found, the enzyme was found in the closed conformation, and the peptide was trapped in the internal cavity with no way to get out, as you can see here. So looking at the internal cavity here, now on the right, you can see with a red sphere is the zinc atom, which is the catalytic site of the enzyme. And as expected, the phosphinic transition state analog is stabilized there. But the peptide extends through the cavity and essentially sits on the lining of the cavity. This is the 10-mer peptide. When we looked at the 15-mer peptide, we had kind of a surprise. Again, on the right side, you can see the catalytic site, and the N-terminus of the peptide is stabilized there. But in the middle, there was very weak density. But then again, we could trace the density of the C-terminus of the peptide reaching out on the other side of the cavity and being stabilized with a particular set of amino acids that I'm going to talk about a little bit more later. This was a different trajectory compared to the other peptide, but again, it was not the only one. We had an alternative conformation that indicated that the middle of the peptide was rather flexible, but the ends were restricted to binding these two sites, the catalytic site and what we call the C-terminus recognition site. So we think that peptides, what we saw was essentially two different binding modes of the peptides. One was for really long peptides that are stabilized from their N-terminus and they're from the C-terminus, and they have some flexibility in the middle that allows the enzyme to accommodate many different sequences. Shorter peptides, and in this particular case, this peptide had a C-terminus that carried a positively charged amino acid, can either not reach the C-terminus site or they don't want to reach because the C-terminus site is positively charged, so they can be accommodated on the lining of the cavity. This length, the difference in length, in distance between the C-terminus site and the catalytic site is very reminiscent of MHC molecules. And it's actually, if you measure MHC molecules, if you look at panel C, most peptides bind with an extended conformation, and for a 9-mer, they reach a distance of about 25 angstroms. If you look at the ERAP1, the distance between those two sites in the closed conformation is a little bit bigger. It's about 30 angstroms, which is about sufficient to accommodate peptides that are 1 or 2 amino acids larger. So we think this is the basis for length selection. We essentially have peptides being grabbed by the ends, and these ends are appropriately spaced, so they can only trim peptides that are a little bit longer than what is necessary for binding to MHC. So just a couple of slides on this C-terminus recognition site. I thought when I first saw it was very interesting because it looked like a carboxypeptidase. So the motif it had a motif of 3 amino acids, a tyrosine, a lysine, and an arginine, and they were positioned at locations almost identical, although there's no sequence identity, with known carboxypeptidases. So what we think we have here, we have an aminopeptidase that has a non-catalytic, also carboxypeptidase type motif that can stabilize the C-terminus of the peptide. And of course, if you block this motif, you lose catalytic activity and efficiency. Also, if you just take now the C-terminus of a peptide, and this is done now with a peptide consisting of 4 amino acids, with the same 4 amino acids as the 15-mer, now you can actually see activation of trimming of small substrates in the catalytic site. This was not obvious how it occurs because these sites are, as I said before, 30 angstroms apart. So you should have a mechanism that this site can activate allosterically the catalytic site. We're still looking into that, but one of my colleagues in Demokritos, Dr. Athanasios Papakyriakou, did an interesting dynamic network analysis of how information can travel from the regulatory, the C-terminus side, to the catalytic side, and did find a couple of pathways that are different and could indicate communication between these two sides that could lead to activation. And this is very recent. Crystallography is a very useful tool, but doesn't always show you what really happens in solution. So in collaboration with Larry Stern's lab at UMass, we've been looking at using a small angle X-ray diffraction to look at what is the structure of this enzyme actually in solution. And it turns out from fitting models that the answer is quite simple. In solution, the enzyme is in the open conformation. However, when you put essentially anything, this is shown here with an inhibitor. This DG13 is one of the prototyping tool inhibitors we developed a few years ago to investigate this enzyme and also to investigate its pharmacological repercussions. So when you add an inhibitor on the catalytic side, the enzyme closes, collapses, and takes something that is at least close to the closed conformation that we see in the crystal structure. So what we understand about the mechanism was recently reinforced by a fortuitous discovery. As I said before, we're interested in the medicinal chemistry and the pharmacological applications of inhibiting this enzyme. So in collaboration with GlaxoSmithKline, we ran a large screening campaign for inhibitors. This is one of the hits. This turns out to be a natural product. This was a very potent inhibitor of trimming of large peptides. However, when we analyze the properties of this compound on a small substrate, one of the small fluorogenic substrates we use routinely to study these enzymes, we saw the opposite. We didn't see inhibition, we saw activation, about 2.5-fold. And this was done by increasing substrate affinity. However, when you use a larger peptide, like a 9-mer shown here, the inhibitor is a normal inhibitor, and it will actually appear to be a competitive inhibitor. So how can that be? How can you have an inhibitor having those sort of opposite functions? This is not very important. This shows that it's active in a cellular assay, that it's active in regulating antigen presentation in cells also. So then we got the crystal structure of this inhibitor, and it turns out that this was located exactly at the C-terminus recognition site, where the peptides go and put their C-terminus, and that was about 30 angstroms away from the catalytic site. So we think that this inhibitor appeared competitive because it blocked the C-terminus binding of large peptides, but it would lead to activation of small peptides by the same way that the large peptides C-terminus does. So just a couple more slides that don't take too much of your time. Just I want to point out with an emerging direction of this enzyme. It turns out that ERAP1 is polymorphic. This is quite common on genes that are important for the immune system and differentiate our responses to pathogens within a population. You can define what we call allotypes, which are combinations of particular polymorphisms within the gene. And turns out with a recent genetic analysis we did that essentially in Europe, 99.9% of the people have one of 10 possible allotypes. These allotypes are defined by particular mutations, common polymorphisms that are scattered all around the enzyme. These are shown here, and they're all away from the catalytic site. So we've been studying these allotypes, and one thing we immediately noticed is that their enzymatic activities vary quite a bit. And actually there are some extremes, some outliers. If you look at the graph on the left, this is a Michaelis-Menten analysis for trimming of a 9-mer peptide. And you can see, for example, allotype 10 seems to be enzymatically defective, at least for this particular substrate. What we also did is map activity and prevalence of these allotypes in the population. On the graph on the right, this is on global population. You can see there is quite a wide distribution. There are common allotypes that are very active, and also some common allotypes that are very inactive. So we think that this is important in driving heterogeneity in immune responses. So we also wanted to make some sort of contribution to understanding the current pandemic. So we use these allotypes to see if there are any difference in generating antigenic peptides from the S1 spike glycoprotein of SARS-CoV-2. And we did see quite a bit of differences, and the patterns were variable. There were some allotypes that clustered together and they were similar, and some were, again, very different. Again, allotype 10, which I need to point out that in Europe is about 5% of people, and about 2% of people are homozygous for this allotype, had very different patterns of antigen generation. And when we even looked at the generation of known antigenic epitopes for SARS-CoV-2, which is shown on the right, again, some epitopes are produced fine from all allotypes, but some require only particular allotypes. And also, if you look on the column of allotype 10, this seems to be more deficient in generating many important immunological epitopes of SARS-CoV-2. So one of the things we think that should be looked at is if there are any correlation between HLA, which are already known, ERA1 allotypes, and tendency to have severe COVID. That's all I wanted to talk about today. I'd like to thank you for your attention. I also thank my collaborators who allowed this work to be performed, and the funding, of course. Thank you very much.